Great. Now I can start recording. Yeah, so আমি actually draw করতেছিলাম আমার so yeah কতটুকু পর্যন্ত তুমি শুনো নাই?
আপু তুমি draw করতেছিলে এখনো you didn't start telling you didn't start labeling the things you just draw করতেছিলে
Last day I asked you, yeah, I was right here, so I'm just gonna point it out right here. So like, do you know what this is? This is a plant cell, right? So
yeah, it's
কি এটা?
chlorophyll.
Exactly, it's a chloroplast. And what does it do?
It has chlorophyll inside.
Exactly,
It helps
has
with photosynthesis.
Yeah. Okay, and then yeah, so I think you saw the labelings. That's a cell wall, the outside, the line that I drew outside, and ভিতরটা হচ্ছে cell surface membrane. So this entire thing right here is a nucleus, and nucleus এর ভিতরে যে strands গুলা, those are chromosomes, and the dense region is nucleolus, and this big hole, this one, this is a vacuole, and this is actually filled with cell sap. Okay, its membrane, the vacuole's membrane is called tonoplast. Okay, now if you are done drawing all these, I can remove them. Actually, wait, this one is a mitochondria. Okay, hello Saiba.
Apple, the mitochondria and chlorophyll, they look the same, just one has color and the other doesn't.
Yeah, exactly, that's why I actually highlighted it with green. They actually look very similar, but yeah, there is a difference obviously. That's why you need to label when you're drawing in the exam. Obviously the examiner wouldn't know যে তুমি কি একটা mitochondrion আঁকছো নাকি একটা chloroplast আঁকছো, so you have to actually label your drawings.
The thing coming out of the nucleus, that's the endoplasmic reticulum as well, right?
Oh yeah, so these are the endoplasmic reticulum. You don't really need to know if it's smooth or rough. আপাতত এতটুকু জানলেই হবে. And by the way, like somewhere else, we also have Golgi apparatus. So it's not like it's absent in plant cell, but yeah, it is still there. Okay.
Yeah. Apu, about the vacuole, in the plant cells, they are temporary, right? It can be there or it can't. But in the in the animal cells, they're temporary, and in the plant cells, they're permanent, right?
Yeah, exactly. So these ones, so animal cell এরকম ছোট হয়, and they can form and they can, not be there. But plant cell, there's always this central big vacuole. Okay. So yeah, also like I think I showed you ribosomes in animal cell, but there are ribosomes in plant cell as well. Okay. Oh yeah, and this is the cytoplasm, obviously, and these dotted things are ribosomes. Okay, once you're done, I can remove this.
No problem. I wrote it.
Okay, great. Okay, now I'm তোমার syllabus দেখলাম, and you actually need to be able to prepare a slide, a microscopic slide, and also you need to be able to identify different what do you say, different cells. So specifically they mentioned these 3 types of cells that you need to be able to um, see under a microscope. So they actually mentioned liver cells, onion epidermis, epidermal cells, and elodia, which is actually a moss intercellula. Okay, so I'm going to show you all 3 of these, and yeah, you need to actually remember them, and if possible like a line drawing kore rakho. Okay, so let's start with, I think, liver cells. Okay, these are the liver cells. Okay, I cannot really. So these are the liver cells. If they tell you to draw a line diagram, like at a paper 6 এ আসতে পারে, even paper 2 তেও আসতে পারে. So all you have to do is just, you know, border type of আঁকবা, and whatever you can see, you just draw them. Extra কিছু draw করা যাবে না. Okay, so like that. Okay, that's it.
Yeah.
I think you already know this is the nucleus. This right here, what is this?
This is the nucleolus.
Exactly, this is the nucleolus, and this is the cell surface membrane. And obviously this is the cytoplasm. That's all you can see under the light microscope. Okay, so that's it, that's enough. All right, if you want you can take a picture of it, or oh yeah, I'll be sending you the recordings, don't worry. Okay, yeah, so we're done with this. Now the next one, which is the onion epidermis, I'm also going to show you that. So next page. So this is actually the cells from onion epidermis. Epidermis means the outer skin, okay, of the onion. So অনিয়নের বাইরে দেখবে একটা পাতলা একটা ছালের মতো থাকে, ওইটাকে যদি তুমি মাইক্রোস্কোপের নিচে দেখো, then you'll be able to see this picture. Okay,
Yeah.
what is wrong with this thing? Okay, yeah, so all the things are already labeled, so you can see the nucleus, the cell wall, the cell membrane, and also the vac wall in the middle. Yeah, the vac wall is not really visible, but maybe you can see some shades, you know, some at a light, at a color দিয়ে একটা কিরকম জানি আছে, so ওইটাই হচ্ছে vac wall আর কি, দেখা যায় না clearly, but that's what it is. Okay, yeah, and then what do we have? Character. This one is the এটা হচ্ছে cells from moss হচ্ছে at a type green like plant. Obviously they don't have flowers, মানে এগুলো কেমন জানি বুশের মতো, মানে মাটির উপরে গ্রো করে. ওকে, and then অনেক স্লিপারি, অনেকটা algae টাইপের. ওকে, So you can easily see that that's the cell wall, and obviously ভিতরটা হচ্ছে cell surface membrane, and these green ones, these are chloroplast.
Apu, if they ask me to draw a diagram, I won't have to draw anything other than these three things, right?
Exactly.
Like whatever I can see from the diagram, I'll just write that.
So এটা most probably paper 6 or maybe paper 2 to আসতে পারে যে draw a line diagram or maybe just draw a cell of Elodia or something. Then all you have to do is just copy this. Like এটা আঁকলেই হবে এভাবে। যা দেখতে পারতেছো তা আঁকলেই হবে। এত ডিটেইলে কিছু আঁকা লাগবে না। So you can see, you cannot even see the nucleus. You can only see the cell wall, the cell surface membrane, and the chloroplast. So এটা আঁকলেই হবে। That's it. Okay, done copying, I think.
Yeah.
Now you need to be actually be able to prepare a microscopic slide. So that's the way. I cannot really zoom in, but I think you can clearly see যে কি কি লেখা. So এখানে they're preparing a slide of the onion skin, onion epidermis. Okay. So you can see that they're peeling off an onion with a tweezer. This is a tweezer. Yeah, it's already labeled, sorry. And then simply বাইরে যে পাতলা skinটা, which is the epidermal tissue, ওটা নিয়ে একটা glass slider উপর drop of water first add করছে, and then এই epidermal টিস্যুটাকে place করা হয়েছে, okay, ওই পানিটার উপরে, and then you put the stain. So stain হচ্ছে এখানে iodine. You could also use methylene blue. So these are the common stains, iodine and methylene blue. Okay, and then basically place the cover slip. Okay, so that is the last step, placing the cover slip. So কি করতেছি, tweezer দিয়ে প্রথমে epidermal টিস্যুটাকে extract করতেছি, and then পানির একটা glass slider উপর আমি প্রথমে পানির drop দিব, and then we place the epidermal tissue into the water, and then we add the stain, and finally we add a cover slip on top. Okay, and then it's ready to view under the microscope.
Apu, if we don't stain it, are we not able to see what's the contents of the cell?
No, because অনেক like epidermal যে টিস্যুটা, epidermal যে সেলটা, ওটার কোনো নিজস্ব কোনো কালার নাই, so you won't be able to really notice the structures. So just to make everything clear, like all the structures and everything of the cell, that's why you need to add the stain. Okay.
Apu, after this, we can see like the diagram you showed me, the
exactly.
Twitter পরে
Yeah.
ওগুলো visible হবে, right?
Exactly. Once তুমি যদি microscope under দেখো, then this first picture, this one,
Yeah.
এটা basically you will be able to see. Okay. Done, I think.
Yeah,
Okay, great. Next হচ্ছে, okay, now will be, I think we need to study different tissues, and yeah, but also different organelle functions gula in detail shikha lagbe. So do I have to go through them? Do you want me to write them down? Naki tumi you already know or you already have those notes?
Yeah.
Hello? Hello? Hello?
Well,
Hello, Saiba? Hello?
hello.
Yeah. So, can you hear me?
Yeah, I can hear you.
Okay. So do I have to write down the organelles and their functions?
Apple would be better because I don't think I have like the defined notes of them.
Okay, great. Then let's write them down. Um, so let's start with the simplest one. Can you see? Hopefully, I'll try to need stick.
Yeah, I can see.
Okay, number one is cell surface membrane. So what does it basically do? Like you said, it's a partially permeable membrane. Surrounding the cytoplasm of the cell. I mean, functions gula blue color ligbo, okay? So it controls the substances entering or leaving the cell. Do you know kichu functions of cell surface membrane? It holds all the organelles and gives the cell its shape. Basically, it acts as a boundary, as a wall. Okay.
Yeah. Apple for
Then
plants, their shape. I thought plants get their shape from their cell wall, and animals don't have a definite shape.
yeah, exactly. Animals don't really have a definite shape. Uh, I would say if you don't want to write it this way, and you can simply say that it just holds all the organelles, and it acts as a boundary. So it's like um yeah, just holds all the organelles would be enough. You're right. It does not really give it a shape. It just holds them together. Okay. So yeah. Yeah, so yeah, the cell surface the animal cell actually does not have a shape. That's a very good point. The plant cell has a rigid shape. Okay. Number two is cytoplasm. Okay, cytoplasm. Cytoplasm is just a gel-like matrix embedded with organelles. Now, what is its function? Oh man. It is the site of most of the cellular activities. So all the cellular activities, they actually happen in the cytoplasm. Okay.
Yeah.
Number 3, we have the cell vacuole. So these are just for animals. These are small fluid-filled spaces bound by a membrane. Basically, they store water and food substance. Not permanent. Okay, what about plants? Yeah, so plants, they are quite large. So large, filled with cell sap and surrounded by tonoplast. Okay. Yeah, so what does cell sap contain? It's just made with water, minerals, and amino acids and stuff like that. I'm not writing it down, okay? Yeah, that's it. Okay, next. Nucleus. Which is an organelle surrounded by nuclear envelope contains nucleolus and chromosomes which contains DNA. So basically the function of nucleus is, oh my God, how did I have that? The function of nucleus is it controls all the activities. What activities such as, cell growth, uh, repair, cell division, these kind of stuff. I don't think you need to know much detail about endoplasmic reticulum and Golgi apertures, so I'm not going to write them down. I'm going to move on to mitochondria and chloroplast then, okay? Do you
Yeah.
think, do you need endoplasmic reticulum and Golgi apertures? I don't think so.
Apu, could you repeat the question again? I couldn't hear you.
Like I actually asked you যে আমার কি Golgi apertures and endoplasmic reticulum লেখা উচিত নাকি should I just skip
Apu,
them?
আমি কোনো question paper like particularly দেখি নাই, like a question about
হ্যাঁ।
Golgi apparatus or endoplasmic reticulum, কোনো क्वेश्चन पेपर आपातत আমি দেখিনি।
Exactly. I'm just, I want to follow your syllabus, and your syllabus does not really mention that. So let's skip it, okay?
Okay.
So mitochondria, mitochondrion. So what is that? These are small elongated organelles with folded inner membranes, aerobic respiration. Oh, this is the function basically. Why am I writing in black? Okay, so aerobic respiration takes place here. That is where energy is generated, you know? So
Yeah.
okay, next is chloroplast. So chloroplast, these are oval, you know, membrane bound organelles filled with chlorophyll. Yeah, function is, these are the sites for photosynthesis. Okay, that's it. I think I missed out to one more thing, that is the cell wall, and then we're done. Are you done writing this part?
So do we need to know about ribosomes, like the functions of ribosomes?
No, I don't think so, because ribosome আরো পরে আসে. So endoplasmic reticulum Golgi is ribosomes. You'll be able to learn all of these in your A levels. Okay.
All right. I'm done taking notes.
Okay, great. So yeah, the last one was cell wall. So cell wall is basically non-living and fully permeable. Not a membrane, like it's a wall. So non-living fully permeable wall. Yeah, that's it. Sorry. So its function is it protects the cell from injury. Gives its shape and maintains the turgidity. Okay, that's it.
Who maintains the turgidity of the cell means that it holds the shape, right? Basically.
Yeah. These are actually the same point. It mainly means that it holds its shape. So there's, সেল যে যখন একটু শুকায় যায়, যখন পানি বের হয়ে যায়, একটু different shape হয়, it's not turgid anymore. But this, because of this cell wall, it can actually be turgid for a very even a very low concentration of water. I mean, even if there's very little water, it can actually be turgid. I think you learned this in like the plant chapter or something like that. But yeah, apadato, I think that's enough.
Apu, I had a question. Photo. They say we need to understand or find out the diagrams from photo micrographs. That means they are from microscopes or something.
Yeah, exactly. So photomicrographs, these are yeah, microscopic photographs basically. One thing I really want to tell you, Jay, if they actually tell you to draw a cell which is seen under light microscope.
Yeah.
So যদি লাইট মাইক্রোস্কোপ মেনশন করে বা তুমি যদি লাইট মাইক্রোস্কোপের একটা পিকচার দেখায়, you have to know যে লাইট মাইক্রোস্কোপে শুধু সেল মেমব্রেন, cytoplasm, nucleus, back wall, cell wall and chloroplastই দেখা যায়। আর কিছু দেখা যায় না। So make sure if they actually tell you to draw a cell which is seen under a light microscope, don't try to draw too many stuff. Don't try to draw your yeah, আমি যেগুলো দেখাইছি একটু আগে, like endoplasmic reticulum, Golgi apertures, nucleolus, তারপর chromosomes, don't try to draw them. এগুলো সব দেখা যায় under electron microscope, and light microscope এ শুধু এগুলো জিনিস দেখা যায়। Okay,
Yeah.
and you'll be able to see because light microscope এর নিচে আসলে বেশি কিছু দেখা যায় না। একটু আগে showed you some photos. These are all under light microscope. That's why you cannot see that many stuff. Okay, for example here, see, you cannot see that many organelles. You can simply see the cell wall, cell membrane, chloroplast or nucleus, এগুলোই দেখা যাচ্ছে, and vacuole, আর কিছু কিন্তু দেখা যায় না। Okay,
Yeah.
so yeah, make sure you know this. That's it. Then we can move on to the adaptation of cells. Are you done copying?
Yeah. Apu, could you also, I couldn't copy like everything, but could you send me the video after the class so I can take a proper note?
Yeah, sure. But I would say, like, আমি যখন class নিতেছি তখনও try to do much of this stuff because yeah, otherwise a lot of people get lazy and they don't really do it afterwards. Okay. যেহেতু
I've
তোমার
taken it, but it looks really rough. So I like after the class, I'll take better notes so I
oh
can
yeah,
understand it more
sure.
on writing.
Yeah, definitely. That's what I want to make a recording. And Alhamdulillah, me Parsi. But there's a downside that this meeting is actually going to end in 40 minutes because apparently I need Zoom Pro to have longer, video calls. But let's see. I'm okay to take that. Um, let's start adaptation of cells. I saw in your syllabus, adaptation or bella, you only need to know about red blood cells, xylem vessels, and I think root hair cells, I think. Yeah. These three are in your syllabus, so I'm gonna go through them. So adaptation, Maniki, adaptation of cells. Adaptation of cells to their functions. Now, not every cell looks the same in our body. Even even in plants, okay, in any microorganism or organism anywhere, সব সেল একই রকম দেখতে না, আমরা সব সময় যে গোল সব স্ট্রাকচার আছে ওরকম ভাবে সব সময় হয় না, okay. So because every cell is adapted to its own functions, ও যেটা carry out করবে, ওর যেটা কাজ সেটা অনুযায়ী or structures গুলা adapted, okay. So the first thing you would learn is red blood cells. Okay. This is the first one. I think you already know how red blood cells look like. They are like, obviously, bhabe dekhle gol lage. But
They
actually,
have a
when you,
biconcave shape, right?
sorry?
They have a biconcave shape.
Exactly. They actually have a biconcave shape. So আমি যদি obviously 3D drawing এ দেখা যায়, but this is a 2D drawing, so it's difficult to show, but I think you get the idea. So actually, when you try to draw a cross section, it will look like this. Okay.
Yeah.
Like, let me try to actually draw it properly so that you can see. মানে ধরো আমি এটাকে মাঝখান দিয়ে কেটে দিছি, ঠিক আছে, কেটে এরকম ভাবে ড্র করছি, so you can see the cross section. It is like dumbbell shaped বলে এটাকে, okay. So this is actually and yeah, this is a biconcave disc shape, okay. So first of all, red blood cells, let me call them RBC in short, okay. আমি বারবার red blood cell লিখতে লিখতে মরে যাব। আচ্ছা, RBC, they do not have any nucleus কিন্তু,